Hello and welcome to the NGS Webinar Series hosted by the Next Generation Sequencing Competence Network. Along the series, you will be hearing from scientists and clinicians from various fields of research. However, they are all connected by sequencing. Who would have guessed? We bring you to journeys where researchers are telling us about their sequencing data harvesting and bioinformatics analysis and surely results and perspectives. How do they do it? What is it good for? Watch and let us know. Our network has started as a DFG-funded national collaborative initiative in 2019, and up to 2023, there have been several annual calls for projects to be sequenced at our core genomic facilities across Germany. From 2024 on, you have a new exciting opportunity to co-apply with one of our genomic facilities for a high-cost sequencing project, which can cost up to 1 million euros. If you ask me, it's worth to check it out. Go for it. We'll leave the link in the footnotes, and here is QR code for your fast access. Welcome aboard. Find a comfortable seat, get a drink, and let's listen to our next speaker, shall we?
Thanks for the invitation and for joining today. I am Anna Esteve, and I'm leading the functional genomics team here at CNAG in Barcelona, Spain. And today I will show the added value of combining RNA-seq with long-read whole genome and methylome sequencing for rare disease diagnosis. And for that, we will walk through real cases where this integration helped with the diagnosis. So let me start with the basics. In Mendelian disease, the cause is a rare DNA variant. So why do many cases stay unsolved after exome or short-read genome sequencing? Because the causal change often sits in non-coding or complex regions, like deep introns, promoter, repeats, etc., places that are hard to see and even harder to interpret without a functional readout. The variant effect can show up in the transcriptome in the form of aberrant expression or aberrant splicing, and or in the epigenome in the form of aberrant DNA methylation. So to capture these effects, we integrate long-read whole genome sequencing that allows us to detect structural variation, resolves repeats and other hard-to-map regions, bases the variants to determine if they are in cis or trans configuration, and directly profiles DNA methylation in a single assay. Whereas RNA-seq allows us to detect aberrant RNA phenotypes. And here the goal is to prioritize overlooked variants by triangulating RNA and or methylation aberrations with nearby rare DNA variants. So, all right, what does each layer actually give us in practice? Let me start with RNA-seq, framed as an outlier detection problem. Here we have to compare our patient against a reference cohort and look for genes that are statistical outliers. For expression outliers, genes whose expression is unusually high or low for that tissue, think, for example, with zeta scores that are from 0, and that flags those such changes like loss of function, copy number variation, promoter or enhancer defects. For splicing outliers, RNA-seq detects junctions with abnormal percent splicing. And that can create, for example, cryptic exons, exon skipping events, intron retention, exon truncation, or exon elongation events. These events often point to deep intronic or regulatory variants that you won't see from coding DNA alone. For RNA-seq can also detect monoallelic expression. These are transcripts. This is when the transcripts come from one allele only. A premature stop codon on the other allele may trigger nonsense-mediated decay, so the mutant allele disappears in the RNA. Finally, RNA-seq detects abnormal RNA fusions. When the genome is rearranged due to, for example, deletions, inversions, duplications, or translocations, the RNA shows the junction that stitch parts of two genes together. So using RNA-seq, we can see what's wrong in the transcript. Whereas long-reads whole genome sequencing resolve complex structural variants, mobile element insertions, repeat expansions or contractions that short reads struggle with. They also face variants across long stretches, so we know if two hits are in cis or in trans, and this is critical for recessive diseases. They are sensitive to mosaic structural changes as well, and because we are sequencing native DNA, we also get the 5-MC methylation in the same run. And with that, we can assess, for example, imprinting defects, non-random X chromosome inactivation patterns, or even to detect episignatures or other methylation aberrations. So here is how we run this in practice. To detect RNA aberrancies, we can use DROP. It's a pipeline that uses Outrider for the detection of sample-specific expression outliers and Fraser to detect splicing outliers. For RNA transcript fusion detection, we use Arriva. And for the long reads, we use the ONT epi2me human variation workflow that calls SNVs, index, copy numbers, structural variants, repeat expansions, and also is able to phase the variants. Because these are native DNA reads, we can also extract that 5-MC methylation. When the phenotype suggests a methylation syndrome, we can submit methylation bed files to the EpiGen Central for a known episignature inspection. And the way that we interpret this is simple. The DNA is the cause, RNA and methylation is the effect. So we can either start from the DNA. For example, if we have a candidate variant, and we ask, do we see the matching RNA or methylation effect in cis? Or we can start from the RNA. We detect first the expression or splicing outliers, and then we look nearby for a DNA change in cis. So let me zoom in on the DNA methylation episignatures. These are syndrome-specific CpG patterns in blood. They give us orthogonal functional evidence. We can support or refute a suspected diagnosis, and in the right context, they help with variants of uncertain significance. As of today, there are more than 100 clinically validated blood episignatures across well over 100 genes and regions. And it's not always a strict one-to-one relation, one gene, one episignature. One gene, one episignature is common. Many chromatin regulator genes, for example, but some genes show subsignatures by variant class, region, severity, or even sex. One locus, multiple episignatures happens in imprinting domains, for example, where the parent of origin flips the episignature pattern. CNV's syndromes, deletion versus duplication of the same region, can yield also different episignatures. But it's also possible that the causal gene have no robust blood episignature detectable. In routine practice, labs run epic arrays and compare the patient to curated reference cohorts using supervised supervector machine classifiers. The output is a classification score and graphics like heat maps or clusterings showing how closely the patient matches a known episignature. It's powerful for phenotypically heterogeneous disorders, and it provides functional evidence that's very handy when genetics alone is ambiguous. But remember that not every condition has a robust blood signature. Results must be integrated with a clinical picture and the genomic findings. Looking ahead, with long-read sequencing, episignature detection in blood is feasible and brings haplotype-aware methylation in the same run. But reference cohorts and external validation are still catching up. For now, arrays remain the front line. So we start with this study, episignatures with long reads. To answer the question, do long-read methylomes reproduce the array episignatures? Here, Geisens et al. Ran a prospective ONT long-read whole genome in 20 patients, covering 13 syndromes with known episignatures and 40 controls. They compared the long-read methylomes to the microarray references and trained per syndrome supervector machine classifiers. The result is that long-read methylation patterns match the array episignatures. Here we can see the hue map and heat maps at signature loci for Sotos, Kabuki, Widermann-Steiner, Cornelia de Lange syndrome, showing that cases cluster with array references, demonstrating the concordance. In this cohort, there was a female with Borgenson-Frostman-Lehmann syndrome due to a de novo PHF6 frameshift. This syndrome is X-linked. Males are typically more severely affected than females, while females show variable expressivity due to X chromosome inactivation. Per read methylation, we can look at each haplotype separately. In her blood, the promoters of these three genes that are located in the X chromosome show a very strong skew. Roughly 70% to 80% methylated on one allele and 2 to 10% on the other, which is exactly what you would expect with skew X inactivation. Other female controls in the cohort, in the bottom, are much more balanced. And in this study, they also saw that a similar imbalance was found in urine and buccal tissue. So phasing shows that her PHF6 frameshift sits on the hypermethylated inactive X chromosome in blood. That sounds a bit counterintuitive because if the mutant X is mostly inactive, why the phenotype? But remember that X inactivation is tissue-specific and incomplete. It only takes a meaningful fraction of mutant active alleles in the relevant tissue, especially in the brain, to produce the syndromic features. In this other study, RNA-seq was crucial here. In case A, we have a newborn screened with positive severe combined immunodeficiency. Follow-up confirmed T-cell lymphopenia. Triosome found a paternally inherited ATM frameshift, so one hit. Fibroblast RNA-seq detected reciprocal in-frame fusions between these two genes, and that pointed to an inversion in the DNA. PacBio long-range PCR plus long-read sequencing breached the intronic breakpoints and showed the inversion is maternally inherited. Together, this yields a compound heterozygous loss of ATM, the paternal frameshift plus the maternal inversion, explaining the phenotype of the patient. In case B, it's a 9-year-old boy with multiple osteochondromas, suggesting hereditary multiple exostosis, which is an autosomal dominant disorder, more often caused by XT1 or XT2 loss of function. He had also co-occurring neuro features that were explained separately by a pathogenic DCX variant. Prior testing was negative, so it was a standard test investigating these two genes, XT1 and XT2, and it was negative. So they performed RNA-seq and they detected an in-frame SAMD12 XT1 fusion, pointing to a structural event at the DNA level. Follow-up confirmed a 0.6 megabase mosaic interstitial deletion on chromosome 8, spanning these two genes, removing the XT1's 5' region that leads to XT1 haploinsufficiency, which is the mechanism for hereditary multiple exostosis. So why earlier test missed it? Because it was a mosaic CNV that was below the routine calling thresholds. Then we have another study. This was a child with classic Kagami-Ogata syndrome, and whole exome and short-read whole genome sequencing were negative. So they did PacBio hi-fi whole genome sequencing on native DNA. And the key finding was a de novo 203 base pair deletion at the MEC3 promoter and also involving exon 1 on the maternal allele. They also saw a biallelic hypermethylation at 2 imprinting control marks, exactly the imprinting pattern of this Kagami-Ogata syndrome. Because the reads are phased, they could show pattern of origin. The deletion sits on the maternal haplotype, while the paternal haplotype is unaffected. The figure below shows that the per-read haplotype resolved methylation tracks the maternal allele is hypermethylated compared to controls. And here, they detected a minimal critical region that aligns with the 203 base pair event with previously reported Kagami-Ogata syndrome deletions. In other words, is the smallest shared deleted segment that, when is lost on the maternal allele, is sufficient to drive the Kagami-Ogata syndrome methylation pattern. So in this other study, they had 2 unresolved patients with Stargardt disease type 1 with 1 hit in prior testing. This is an autosomal recessive ABCA4 macular dystrophy. where 15 to 20% of the clinically clear cases remain with one or no ABCA4 variant. Then they use short-read whole genome sequence to find the second allele, and long-read RNA, in this case long-read RNA, in retinal organoids to show the functional effect. So, since ABCA4 is only expressed in the retina. What they found is, for patient one heterozygous deep intronic variant, splice activating, and a common hypomorphic missense present in both alleles. Hypomorphic is a partial loss of function allele, contributes to disease mainly in combination with a severe allele. And patient 2, the same intronic splice activating variant in cis with a common hypomorphic missense on one allele plus a rare hypomorphic missense on the other allele. So here, long-read RNA-seq, long-read RNA sequencing in patient-derived retinal organoids demonstrated a retina-specific exon skipping consistent with the intronic variant. Also, in particular, for patient 2, full-length reads were biased toward the haplotype with the common hypomorphic missense. The haplotype carrying the rare hypomorphic missense was underrepresented, which means a reduced stability of the transcript. So the bottom line here is whole genome sequencing supplied the second allele, the second hit. The intronic change is the splicing cause, and long-read RNA-seq in the relevant tissue provided the effect, completing a biallelic explanation for both cases. In this study, they used long-read to face compound heterozygotes. And here a quick intro into the T10 gene. The T10 gene is the largest human gene, hundreds of exons, giant introns, and tissue-specific isoforms. So short reads often struggle with phasing and mapping across its long intronic deserts. In titinopathies, knowing whether two frameships are in cis or in trans is everything for interpretation. This case was a patient with two titin frameships about 100 kb apart. Short-read sequencing detected both variants, but couldn't phase them. So they didn't know if they sat on the same chromosome or on opposite ones. So with PacBio Hi-Fi, they built long haplotype blocks that span a big chunk of the titin gene that goes, in this case, this block from exon 169 to exon 358. So we can see that the maternal frameshift is in the proband and the mother, and it's absent in the father, and the de novo frameshifts appears only in the proband, and both parents are referenced. So the haplotype Hi-Fi reads across the 100 kb show one haplotype carries a duplication and the other haplotype carries a deletion. So they were in trans. So put together, the trio inheritance plus long-read phasing can explain the phenotype as a compound heterozygous titin loss of function, something that short reads alone couldn't resolve. This is an XP22.3 contiguous gene deletion syndrome. The patient is a girl with developmental delay and dysmorphic features. Prior DNA testing showed a 10 megabase deletion on this region, on the paternal X chromosome. but they didn't know which X was active in blood, so the clinical impact was unclear. With RNA-seq, they look at allele-specific expression across heterozygous X-linked SNPs and use the parents to assign the origin. Across the chromosome, the expressed SNPs were monoallelic and paternal. The maternal allele is absent in the RNA. In other words, blood shows complete skewing towards the paternal X. Because the active X in blood is the paternal X carrying the deletion, the genes inside that interval are functionally null in this tissue, no transcripts from the deleted segments. That pattern supports the diagnosis of this XP22.3 deletion syndrome, which is a contiguous gene disorder, and it explains why the phenotype is present in a female, because we have skew X inactivation and is activated the deleted X in blood. In this other study, we have two siblings that were clinically suspected for PI3K delta syndrome, APDS1, which is an autosomal dominant inborn error of immunity caused by gain of function variants in PIK3CD gene. Both showed lymphoproliferation and recurrent infections, while both parents were unaffected, raising the possibility of a de novo or gonosomal mosaic variant. So to test the recurrence risk, they did ultra-deep targeted long-read whole genome sequence in the quartets around the region, and they detected that both children carry the risk haplotype with this variant, the causal variant, and in the father, you can see low fraction mutant reads on the same haplotype below Sanger and short-read detection, consistent with paternal gonadal mosaicism. The table beneath summarizes nearby SNPs. Phased inheritance places the children's mutant allele on the paternal haplotype. Across additional families, the same approach supported maternal gonadal mosaicism in some pedigrees, and the bottom line is deep sequencing with long reads. Let us see low-level parental mosaicism converting an aberrant de novo variant into a mosaic parent and clarifying the recurrence risk for counseling. In this other study, this family shows a classic autosomal dominant familial adenomatous polyposis pattern. Multiple relatives with hundreds of colorectal adenomas and early colorectal cancer risk. Prior panel and whole-exome testing was inconclusive for some affected members. Short-read whole genome sequencing hinted an intronic event in APC gene, but couldn't resolve it. They moved to PacBio long-read whole genome sequencing and could map a 6 kb full-length line 1 insertion in intron 7 end-to-end with clear breakpoints. They then asked for the functional effect. Targeted RNA analysis showed 103 base pair pseudo-exon spliced between exon 7 and 8, introducing a premature stop and triggering NMD. The variant segregates with disease across the affected relative, establishing the molecular diagnosis and enabling cascade testing and personalized surveillance management for the whole family. In this case, it was unrelated males with syndromic features, including intellectual disability, developmental delay, liver disease, and recurrent infections, and had inconclusive whole exome sequencing. They perform RNA-seq from fibroblasts, and ATP6AP1 gene jumped out as the top underexpressed outlier. Here you can see in the volcano and the rank plots. And the sashimi tracks showed aberrant splicing, inclusion of two partially overlapping cryptic exons between exons 2 and 3. Whole genome sequencing found two deep intronic variants that create splice motif near exon 2 and 3 and lead to cryptic exon inclusion. Orthogonal confirmation with qPCR and mini-gene assays and other biochemical assays were consistent with the ATP6AP1 dysfunction. In this other study was a child with global developmental delay, still undiagnosed after standard short-risk testing. They applied PacBio high-field genome sequencing on blood, and they performed a genome-wide scan for rare hypermethylation outliers, and they detected a promoter proximal block at gene DIP2B in one allele. Close to that gene, there were a hypermethylated region. High-field reads resolved LR's CGG expansion, but was between 280 and 300 repeats and falls within the pathogenic range, which is greater than 250. And later, this was confirmed with triplet repeat PCR. So other core samples with smaller DIP2B expansions lack the hypermethylation, supporting the pathogenic threshold. And this is the last case that I'm going to show. It's a patient with a phenotype consistent with myotonic dystrophy type 1, DM1. Prior testing, panel and exome was not informative. They performed triplet prime PCR, and it flagged a CTG expansion, but they couldn't size it. With native DNA long sequencing, they expand the DMPK3 UTR CTG repeat end-to-end. They quantified the size distribution, showing a clear somatic mosaicism, phased the expanded allele, and read the 5MC methylation across the DMPK promoter. The mosaicism evidence is because we have the per read CTG sizes, and they showed a broad and multimodal distribution, often hundreds of repeats wide, not a single peak, and this is a hallmark for somatic mosaicism in DM1. The result is a pathogenic range CTG expansion in the DMPK3 UTR with haplotype resolve, promoter hypermethylation at the expanded allele, consistent with allelic repression. So this table summarizes the diagnostic uplift achieved by adding RNA-seq and or long-read whole genome sequencing prior testing. Rates vary widely across studies and are not directly comparable for the following reasons. So why diagnostic rates vary across studies? First, the cohort composition. This is the biggest factor. If you focus on neuromuscular muscle cohort, you are sampling the right tissue and the genes are highly expressed and the splicing defects are common. So here RNA-seq adds 30 to 35% in many series. In contrast, heterogeneous blood-based cohorts capture fewer disease-relevant genes, so the uplift is more modest, typically between 5 to 15%. Case preselection also matters. Hard cohorts, patients already exhaustively tested with long negative odysseys, naturally leave fewer solvable events for long reads or RNA-seq, so the incremental gain looks smaller. On the contrary, naive cohorts, panel only or early in the odyssey, show higher apparent benefit. The baseline also matters. Was exome or genome already negative, or were some cases only panel tested? And are we counting solves beyond prior genome sequencing, a true add-on, or instead of genome sequencing? So different baselines inflate or deflate the reported uplift. The tissue choice and disease biology. So when you get the best yield is when you assay the pathology relevant tissue. Muscle for NMD, fibroblasts for many mitochondrial or metabolic conditions. Blood frequently misses tissue-specific events, so the yields drop. Also, it matters the definition of diagnosis, because some groups only count pathogenic or likely pathogenic variants. Others also include strong candidates or research-grade solves that will inflate the reported rate. Requirements for segregation or functional proof also vary. Clinical labs often demand them, research studies may not. Also, diagnostic yield tracks with how much you analyze. On RNA-seq, it's ideal to run aberrant expression, aberrant splicing, monoallelic expression, plus fusion detection. Also, a simple Z-score approximation would perform worse than a more sophisticated statistical approach like Fraser or Outrider. On long-read genomes, adding repeat expansion or mobile element insertions, phasing, and methylation on top of SNVs and structural variants is also better for not missing any event. Also, the outlier detection lives or dies with the reference cohort. Sample size and batch effects are very important. With poorly matched or too small controls, we have fewer true outliers and more noise. Regarding technology, on long reads, the platform ONT versus HiFi, or the coverage, or the library approach, ultra-long, adaptive sampling, etc., all change what you can see, especially for structural variants and methylation. On RNA-seq, poly-A versus ribodepletion protocols, read length, strandness, or the RNA degradation affect gene isoform detection and splicing sensitivity. Also, the reference genome matters. If you use AG38 or the T2T or the pan-genome reference, it makes a difference. And so do for the colors, a robust structural color or a robust RNA outlier color is better. Also, having tree information and phasing can make a difference. Also, the interpretation framework. So applying ACMG rules, checking gene disease validity, and having access to functional assays like mini genes, enzyme tests, determines what crosses the line to a clinical diagnosis versus a candidate. And finally, ascertainment and publication choices. Some cohorts are enriched for cases that long reads are great at. For example, a cohort with already suspected structural variants. So if you pre-select those cases, the long read whole genome sequencing hit rate looks higher than it would in an unselected scenario. So this is the playbook for what we should order. Perform RNA-seq when exome or genome is negative and you suspect for a splicing or regulatory effect, or when you have a candidate gene or variant without functional evidence. And remember to pick the right tissue. Long read whole genome sequencing. Choose it when you have short read whole exome or whole genome sequencing is negative, but the phenotype hints at structure or repeat expansion disease. It's also the right tool when phasing is needed, or when you suspect imprinting or epivariants and one native DNA methylation in the same run. But the best combo, if possible, is do proven RNA-seq plus trio long read whole genome sequencing. If blood is uninformative, add a disease-relevant tissue. The goal is simple. Let DNA show the cause and RNA methylations show the effect. Some limitation. One key limitation is the tissue mismatch. RNA and methylation effects are highly tissue specific. So if blood is silent, we need a different source. The rules of thumb are these ones. If it's a neuromuscular disease, go straight to muscle. If a biopsy is unfeasible, use fibroblasts as a backup. Mitochondrial and metabolic connective tissue or heterogeneous rare disease, start with fibroblasts. They are practical to culture, and blood often lacks expression of the relevant genes. For primary immunodeficiencies or immune dysregulation, use PBMCs or blood if PBMCs aren't possible. And for central nervous system, retina, heart, liver genes that aren't expressed in blood or fibroblasts, confirm in iPSC-derived models, neurons, retinal organoids, cardiomyocytes, or hepatocytes. The bottom line is pick the tissue that expresses the genes you care about. When blood does not show the effect, plan for fibroblasts or disease-relevant tissue, and reserve iPSC models for the priority cases where the tissue access is impossible. Another limitation is interpretation burden. An outlier isn't automatically causal. We prioritize by gene constraint, inheritance, HPO match, network or pathway context, and critically cis evidence, phasing DNA with the RNA methylation effect. Use segregation and functional assays when needed. Also, the long-read whole genome sequencing is still expensive and slower. Start with enriched cohorts, use targeted long reads with, for example, adaptive sampling when whole genome is unfeasible. Trios improve phasing and speed interpretation. And finally, episignatures are not universal. Many disorders lack a robust episignature, and a treatment or mosaicism can blur the signals. Arrays remain the most validated. Long-read methylomes are promising, but not yet universal. And treat borderline matches cautiously and corroborate with genetics. So before closing, a word on collaboration. Within the Solve4D project, I helped design and run solvathons. These are intensive 2 to 3 days workshops where clinicians, geneticists, and bioinformaticians sit together to push unsolved cases using multi-omic approaches. This allows us to deliver rapid diagnosis in real time. We are now extending this model through Herdera to sustain solvatons and harmonize pipelines across Europe. More recently, at the UNICAS hackathon in Murcia, we solved 5 out of 12 difficult RD cases. If you would like to join, submit cases, or co-host an event, let's team up and shorten the diagnostic odyssey together. Thanks.